EMBO e-talk. My name is Alejo. I work at the CNIO in Madrid. Before I start with the introduction to Guadalupe Sabio's talk, I'd like to say just a couple of technical things. So I'm going to be handling all the questions. You are able to, by clicking on the Q&A button at the bottom right of the screen, you can write your question while Guada is speaking, and then I'll handle the questions myself. So you can actually vote for questions if you want whatever question you find interesting to actually get it, I think, on the top of the list. So I'll be handling the questions, but you can type them as well as speaking, as I said. And that's for the first part of the session, which will be sort of focused on scientific questions. There's a second part for where the atmosphere is a little bit more relaxed. Not that this is tense, but that everyone will be able to share audio and the video. And that idea is originally built for students, but anyone can participate so that you can ask questions related with the talk and related with whatever you want to ask Guadalupe about her work or about science or whatever you feel. That one you're going to ask directly. I don't think I'm going to handle those, but that's the second part of this. But for now, just the questions go to this Q&A tab, okay? And then I'm going to share my screen before I let Wada to get on the stage. I don't pretend to make an introduction on the stress kinases because I think Wada's going to do that better than myself, of course. Just a few basic concepts on stress. We can define stress as any challenge that basically can compromise or can be perceived as compromising homeostasis, or this sort of dynamic equilibrium that occurs in our cells and in our organs. So when there's a given stressor, of course, there's an opposite response. That the idea of this response is to return the equilibrium to this sort of homeostatic balance, and it involves the detection, the transmission, and all the cellular reactions that basically counteract the disturbance. And of course, this response here is gonna only end once the stressor is over and once the equilibrium is reassumed. And this might happen or not, depending on things that we'll see in my intro and more so in Guadalupe's talk. So the equilibrium might be resumed, but of course, if we think the multiple stressors, both intrinsic and extrinsic, that operate in a cell, not only that they are multiple and impact at many different levels in the cell, also they are interconnected, of course. This is an impossible task for a cell, and it's always adjusting like a rheostat the multiple stressors and the multiple inputs that are inflicted to the cell, both, as I say, intrinsically and extrinsically. An important point is that our stress responses, I think, of course, they have been selected through evolution, so they're fixed to such an extent that things that we take for granted are actually stress responses, just like the circadian rhythm is a prime example of these. And here I borrowed a nice graph from a review by Russell and Lightman, in which what we are seeing here is the fluctuation in cortisol levels through a 24-hour period in healthy volunteers. And we can see the concentration of ACTH and the concentration of cortisol. And a couple of things are important here regarding with the sort of the program stress response. The first one is that you can see a series of pulsatile increases in ACTH that what it does is to trigger the release of cortisol. And cortisol, a glucocorticoid, basically adjusts behavior and adjusts metabolism to the requirements that of course shift throughout the day. And if we see here in this on the average of cortisol levels, we can see that they are programmed to increase slowly and peak early in the morning and then wane throughout the day. And of course, this is a stress response. Actually, peaks of cortisol are more than frequent, but the take-home message from this is that it doesn't mean that that stress response is necessarily unexpected. And another important point of a stress response is that might actually result in a sort of an improved dynamic equilibrium. This is not necessarily a damage control, and there are plenty of examples, quite prominent examples, including the moderate DNA damage and the moderate DNA damage response that might sort of oil the mechanisms to preserve DNA integrity, and the same applies to small and moderate levels of reactive oxygen species and the control of mitohormesis. And caloric restriction itself, through the several stress responses that it triggers, sort of maintains the soma at the expense of other biological functions. So there might be a sort of a positive effect of the stress response that would resume the dynamic equilibrium at a better place. But this is likely not what Guadalupe is going to be talking about, but we have plenty of examples and are quite relevant for human disease of actually maladaptive stress responses as drivers of human disease. And we can think of this as an excess in the magnitude or an excess in the duration of these stresses and these stress responses that actually are more pathologic perhaps than the stress itself. It's difficult to define what is an excess in duration of magnitude, but we can think of this as presumably something that has not been tuned through evolution. And examples of these are very obvious, I think. One of them is sort of the stress response to sort of the limiting nutrient availability, the sort of the stress response that would self-eliminate these cells that have a dangerous malfunctioning, preserving sort of the organism by sort of eliminating this dangerous cell. The response to foreign pathogens, a threatening pathogen in the body, and the response to predators or dehydration. And all these have maladaptive outcomes. And one prime example of this that will be covered by Guadalupe is the development of obesity and metabolic syndrome. But we can think of an excessive elimination of our own cells, and this particularly takes place during aging, that sort of impairs the ability of our body to sort of replenish these organs that are, these tissues that are sort of being teared away. Foreign pathogens, if there's a response to foreign pathogens and the pathogens are not here, it might lead to cell reactivity, and we know that happens. The absence of predators, but a response to fear might generate anxiety, and the preservation of water and electrolytes can drive hypertension. With these examples, and I know Guadalupe will be mostly talking about the first one, but not necessarily, not exclusively the first one. I would like to now switch. I'm going to stop sharing, and I would like now to introduce Guadalupe Sabio. So Guadalupe is from Extremadura, originally in the west of Spain. She studied veterinary medicine in the University of Extremadura. She graduated in 2000 and then conducted her PhD directed by Ana Cuenda in University of Extremadura and MRC in Dundee. And back in those days, she already became interested in stress kinases. And particularly during her thesis, she identified a couple of new substrates of the P38 kinases. She had a strong focus in biochemistry at that point, and but then she decided to switch into to maintain her focus on stress kinases, but she provided a sort of pathophysiological insight. She moved in 2005 to Roger Davis' lab in UMass in Worcester, and she did a stellar postdoctoral phase with a seminal work in using mouse genetics to try to understand the pathophysiological consequences of the deregulation of the stress kinases. After that successful postdoctoral period, she returned to Spain. She set up her lab at the Spanish National Cardiovascular Research Center in 2011, thanks to an ERC starting grant. And she continued to work in trying to understand the physiological and pathological metabolic consequences of deregulating the stress kinases. She will tell us a few of these lessons that we've learned over the years, thanks to her work, and actually has it's been very, very productive and very insightful, and that earned her a few recognitions and awards. I'm not going to name them all because there are too many, but the L'Oréal UNESCO Award, the Princess of Girona Award, Fundación Jesús Serra Award, AstraZeneca for Young Investigators, just to mention a few. And I also have to say that Guadalupe is an amazing collaborator, constructive and resourceful, and has probably the best feature of a collaborator that will return your emails in a minute. Before you actually click the send button, you will already get a response. So without further delay, I would like to welcome Guadalupe on stage.
Thank you, Alejo. Thank you, Gerling and Rita, for organizing this amazing talk. I think it's a opportunity for the young investigator to show our research and to do a little more of network. Thank you, Alejo, for this incredible introduction. I think you summarize the real idea of what stress do, how stress kinases act, and why they have a real, it's important to control their activation. I want to, I will start saying thanks to the people of my lab. Most of the pictures and slides were done for them. I think a lot from Ivana and Rafa. I am very bad picture making, but the main problem of the picture was that it's from all times when we can be together in the lab without masks. We have to do another photo to put all the new people also in the picture. I am going to talk about stress kinases in the cardiometabolic diseases, and why the malfunction of these kinases or hyperactivation of these kinases could arrive to cardiometabolic disease. The beginning start when it was described that stress activated kinases were activated by several stresses, including cytokines, free fatty acids, glucose, oxidant reactive species, ER stress. All these factors are common in obesity, and it was described that GNK were activated constantly in the tissue of obese patient, suggesting that this altered hyperactivation or constant activation of this GNK pathway was deleterious of what and was triggering part of the comorbidities of obesity. Several work have been done to try to understand the function of these stress kinases. These stress kinases are divided into families: the GNK family, that is mainly activated by NKK4 and NKK7, and the P38 family, that is activated by NKK3 and NKK6. While for a long time have been thought that activation of GNK is all common, that all the GNK can have the same function, or the P38 have the same function, nowadays we know that this is not true. One P38 member could have opposite function for another P38 member, and that gives the more complex idea of this stress kinases function in the cell. Once they are activated after stress, they are going to phosphorylate several substrates that are going to help the cell to the adaptation. However, alteration in the phosphorylation or in their activity are going to be important mediator of this deleterious function, for example, death or proliferation in cancer. However, their activation is also important for the differentiation of the cell and also the survival, meaning that a correct function of these kinases is important for the correct homeostasis in the cell. As I mentioned, this physiological activation normally is acute and have to be deactivated quickly with phosphatase or degradation, but there are several pathologies in which this activation is maintained, and one of several of these stress kinases stay phosphorylated with the time in the triggering obesity comorbidities. And this activation have been shown in different tissues, in myeloid cell, in macrophages and neutrophils, in brain, in the adipose tissue, in the liver, in muscle, heart, and pancreas. And although they alteration in their activity could arise secondary disease, their correct activation in this tissue is important to maintain the tissue homeostasis. In the central nervous system, GNK have been shown to be a very important role. It's going to control the TSH T3 excess, but also is going to control the hyperphagy with the GNK1 inducing hyperphagy. However, GNK3 is an exogenous, indicating that while deletion or inhibition of GNK1 will decrease food intake, depletion of GNK3 will increase food intake. And that is one of the examples in which one of the kinases have opposite role to the sister kinase. In the adipose tissue, P38 have been shown to be very, very important in the BAT activation, the brown adipose tissue activation, but also in the transformation of white adipose tissue to brown adipose tissue. GNK have been shown to be important regulator of some of the adipokines and cytokines that are expressed during in white adipose tissue. Some of these adipokines, like adiponectin, are inhibited by the activation of GNK1, and some of these adipokines, for example, interleukin-6, is activated by the activation of GNK1. In the muscle, it has been described that hyperactivation of JAN is going to induce the iris phosphorylation and inhibition of insulin sensibility in the muscle. However, P38 are very important in the contractibility and in the regeneration of this muscle. And for that, again, it's very important good control of the activation of these kinases for homeostasis. I am going to stop a little more about what we know of the P38 in heart. It has been described from the beginning that P38 were important in the development of the cardiomyocyte, and that inhibition of P38 alpha will activate cardiomyocyte development or differentiation. That this was thought that for inhibition of P38 alpha will help to the cardiomyocyte regeneration. And this was demonstrated in zebrafish, for example, that inhibiting P38 alpha will activate the regeneration of this heart. In the other hand, P38 beta also have a very important role in the heart development. It has been shown that a deletion of P38 alpha and beta in the cardiomyocyte have a cumulative effect, and still is not very well known what is the function of this P38 beta alone. In the beginning, the most studied model of cardiac stress was the ischemia perfusion, and they were studies using P38 inhibitor SB203580 that is going to inhibit only P38 alpha and beta, that demonstrate that inhibiting P38 alpha and beta will induce pro-survival effect of the cardiomyocytes, and that P38 alpha will have a very important role in the mitochondria and controlling not only the cardiac fibrosis after ischemia perfusion, but also inducing cardiac arrhythmias. Moreover, in the thoracic aortic constriction, in this model of heart injury, it was shown that MLKs, that is upstream activator of GNK and P38, is important in the fibrosis of these cardiomyocytes. And in this effect is very important the activation of GNK, and inhibition of MLKs will protect these mice against the fibrosis in thoracic aortic constriction. Moreover, it was shown that P38 activation is going to induce the expression of TNF-alpha and interleukin-6 by cardiomyocytes, and also, very importantly, TGF-beta in the myofibrils that are going to induce also cardiac fibrosis. For that was thought that inhibition of this pathway, GNK and P38, in cardiac damage will be beneficial for the treatment. Moreover, it was shown that GNK2 is a very important regulator of the calcium leak in the sarcopenic reticulum. GNK2 is going to phosphorylate calcium-calmodulin kinase 2 that is going to phosphorylate renin receptor 2, inducing calcium leak and inducing arrhythmias in these cardiomyocytes. As I mentioned before, it has been described that P38 are important in the cardiomyocyte differentiation, but also in the cardiomyocyte proliferation, because both effects are very tight regulated. P38 alpha inhibition have been shown to induce cardiomyocyte proliferation through increased phosphorylation of RB and expression of some of the important cell cycle regulation in these cardiomyocytes. Recently, we have described trying to understand what was the function of the other P38 kinases, that P38 kinase gamma and delta were important in the cardiomyocyte hypertrophic growth, because P38 gamma and P38 delta will phosphorylate Deptor, that is an mTOR inhibitor. Once Deptor is phosphorylated, is degraded, and mTOR is released and activated, inducing protein synthesis and cell growth. In this way, P38 gamma and delta phosphorylation and activation will induce cell growth, hypertrophic growth of the cardiomyocyte, not only in pathological conditions, for example, after hypertension, but also in physiological conditions during the heart development. And doing this experiment, we realized that the expression of these two kinases, P38 gamma and P38 delta, was not present during the early postnatal period and start to be present near the metabolic suite of the cardiomyocyte through from a hyperplastic cardiomyocyte to a hypertrophic cardiomyocyte, or the metabolic suite from the glycolytic metabolism to oxidative metabolism. And we decide to check what was the function of these two kinases in these metabolic suites. We know that in the postnatal cardiomyocyte, in the early postnatal development, the cardiomyocyte needs glycogen as a source of energy, and this glycogen is produced by the glycogen synthase 1, that is an enzyme that is going to be inhibited by GSK3. Later on, in the adult cardiomyocyte, glycogen synthase 1 is inhibited and is not producing so much glycogen, and the cardiomyocytes are not going to need this glycogen as a source of energy, and are going to be more are going to oxidate lipids. This metabolic switch is going to appear between day 7 and day 14 in the mouse cardiomyocytes, and is very important, this switch to the correct function of the heart. And we found that P38 gamma and P38 delta are activated in this moment when the cardiometabolic switch appear after birth. And we found that P38 gamma and P38 delta are going to phosphorylate glycogen synthase 1. This site of phosphorylation is a priming site for GSK3. It's a phosphorylation site that is going to help GSK3 to bind to glycogen synthase. And once GSK3 is bind to glycogen synthase, is going to phosphorylate glycogen synthase and inhibit the activity of this enzyme, inhibiting the glycogen production. And we found that if we alter this function of the P38 gamma and delta, the development of this cardiomyocyte is going to be compromised. And not only this cardiomyocyte, also the crosstalk between the cardiomyocyte and the other organ are going to be very important and are going to be altered. The next metabolic tissue that we are going to talk a little more is the liver. Liver is one of the main controllers of the whole body metabolism because it's going to control lipids and glucose metabolism. And in the liver, it has been described several functions for GNKs and P38, the stress kinases. Both kinases have been described to control these two main functions of the liver, the glucose metabolism and the lipid metabolism. But not only that, the activation of the stress kinases in the other tissues, for example, adipose tissue, central nervous system, or myeloid cell, are going to affect the progression of the steatosis, the non-alcoholic fatty liver disease that appear during obesity. That means we are going to have a function of the stress kinases in the hepatocyte, but also in other cells that are going to communicate with this hepatocyte and are going to be important regulator of the progression of the non-alcoholic fatty liver disease. For example, in myeloid cells, it has been described that GNK and P38 are important regulators of the activation of this myeloid cell that are going to promote the expression of cytokine like TNF, interleukin-6, and also that P38 are going to be very important in the movement of these neutrophils to the liver. These neutrophils are the first effector in the non-alcoholic fatty liver disease, are going to mediate the initial inflammation and are going to also ask macrophage to arrive. That means this initial infiltration of neutrophils is going to be very important for the progression of the disease. How is this non-alcoholic fatty liver disease? It's a disease that is progressive, that is very difficult to diagnostic, and that in the beginning we have a liver that is going to accumulate fat. These lipids in the liver are going to induce inflammation and is going to start to induce also fibrosis. When the fatty liver progress to non-alcoholic steatohepatitis that have an increase of fibrosis, then we can think that the disease is quite serious, because that will progress to cirrhosis in a 15-25% and to ACC, to hepatocellular carcinoma in a 7% of the cases. And once you pass to cirrhosis, this going back is not possible. However, if we are able to stop the patient in the NASH or in the fatty liver, we can reverse the disease if we are triggering the correct signalings. And in this progression, the function of the P38 and GNK are very important in several of the steps of the disease. It's known that the non-alcoholic fatty liver disease is going to start with the insulin resistance that is going to appear first in the fat. This insulin resistance in the fat is going to increase inflammation and lipolysis. This lipolysis is going to increase the free fatty acids that will arrive to the liver, and it will promote increase of the accumulation of lipids in the hepatocyte that will get a metabolic stress, because it will need to metabolize this excess of lipids that it will induce lipotoxicity. This will finally end in death of some of these hepatocytes that will produce increased inflammation, activation of the repair to try to increase the proliferation of the remaining hepatocyte to make that this liver will function normally again. And with that, also the activation of the stellate cell and the increase of fibrosis. In all this cycle, that is a cycle of this disease, the stress activated kinases are very important. In the adipocytes, activation of the stress kinases are going to be important, not only inducing this adipocyte induced insulin resistance and increasing the lipolysis, they are also going to alter the cytokine secretion of this adipocyte. And for example, we demonstrate that the expression of interleukin-6 by activation of GNK1 in the adipocyte was enough to induce liver steatosis and insulin resistance in the hepatocyte. This demonstrates that activation of a stress kinases in the adipose tissue will affect the physiology of distant tissues like the hepatocyte. Moreover, when the hepatocytes are going to receive this signal, they are going to increase this lipid oxidation, but also they are going to increase the accumulation of lipid in this hepatocyte. It has been described that JNK and P38 activation in this hepatocyte are going to increase steatosis by different mechanisms. For example, activation of JNK is going to inhibit fatty acid oxidation, increasing then the lipogenesis, and also is going to inhibit the expression of FGF21, that is an important hepatokine that is going to protect the liver against the steatosis. Moreover, JNK activation are going to induce also cell death in these hepatocytes. Once we know that stress activation or alteration of stress activation in the hepatocyte increase the disease in this model, also inflammation that will occur after that is also controlled by stress kinases. As I mentioned before, activation of stress kinases in monocyte, Kupffer cell, and also in neutrophil are going to be important in the infiltration of this cell to the liver, and also in the activation of this cell in the liver. It has been described that during the obesity and the non-alcoholic fatty liver disease, the Kupffer cell are going to be replaced by this circulating monocyte that will differentiate to a resident Kupffer cell from monocyte. And these new cells are going to be more inflammatory than the initial Kupffer cell. And we are studying how stress kinases are important modulator of this differentiation. As I mentioned, also during the activation of this inflammation, LPS is going to be one of the triggering, also the increase of interleukin 6 and other cytokines are going to activate these stress kinases in macrophage and neutrophil and are going to infiltrate in the liver, increasing the inflammation of this liver and promoting the progression of the disease. An important factor in the non-alcoholic fatty liver disease have been shown to be the gut. During this non-fatty liver disease is going to increase the permeability of the gut, and this will increase the lipopolysaccharides that arrive to the cells in the liver, not only the hepatocytes, but also the Kupffer cells and the hepatic stellate cell. And LPS is an important activator of stress kinases, GNK and P38, and have been described the activation of GNKs and P38, but LPS are going to induce hepatocyte damage, increase of the inflammatory cytokine by Kupffer cell, and also increase of fibrosis by stellate cells. Moreover, recently it has been described that the imidazole propionate that is produced by the microbiome activate in the liver P38 gamma. This activation of P38 gamma is going to activate mTOR1 that is going to inhibit the insulin receptor signaling, inducing insulin resistance in the hepatocytes. Not only that, it was described that P38 gamma is able to phosphorylate in AKT, and AKT is going to phosphorylate AMP in the non-canonical site that inhibit the phosphorylation of AMPK that appear during metformin stimuli in the threonine 172. These two effects are going to induce insulin resistance in the liver. After inflammation, reparation, we have the try to regenerate the damaged hepatocyte. And this regeneration of the hepatocyte that die during this process are going to induce the liver cancer. And in this liver cancer have been described, the activation of P38 gamma is going to be important in the cell cycle initiation in these hepatocytes. Moreover, it has been described that activation of GNK in myeloid compartment also is going to induce ACC, and that activation of GNK in the adipose tissue can also increase the predisposition to liver cancer. However, as I mentioned from the beginning, sometimes these signals have to be in the correct way. And for example, we have described that deletion of GNK1 and GNK2 in the hepatocyte to try to avoid the damage of this hyperactivation of GNK1 and GNK2 during the obesity. However, this inhibition of these kinases is going to produce cholangiocarcinoma because these hepatocytes are not able to have a normal metabolic profile. And we think part of this is because these young have a very important role in the liver circadian regulation and have been shown that alteration in the liver circadian regulation can induce different liver cancers. And we describe that GNK have a circadian regulation of activation in the hepatocyte, and this activation of GNK in hepatocyte is going to be very important in the circadian regulation of the metabolism of this hepatocyte, and that alteration of this signal is going to induce liver steatosis. As I mentioned, stress kinases are not only important in one tissue in the body, are very important in several tissues, and that make very important to try to understand the crosstalk between them, to try to use them as a metabolic target to treat obesity and its comorbidities, because we need to understand what is the function and needs of the tissues. And I hope that I show to you that they are a yin and yang part in the activation of P38 and GNK. They are important in the development, and they are important regulating homeostasis of the body. However, they are going to be its alteration are going to induce several diseases like diabetes, insulin resistant, non-alcoholic fatty liver disease, cardiovascular disease, cancer, immune disease, and central nervous system disease, and we need to know more about them. And I left you now with a video of what we are doing, of how the lab is moving, and how we think the crosstalk between organs is very important.
Unhealthy food, sedentary life, genetic factors, and alteration in the circadian rhythm affect the wellness and drive the dysregulation of our adipose tissue. In the adipocyte, all these factors are going to disturb the adipocyte internal clock and the organelle that produce the energy, the mitochondria. Dysfunctional adipocytes are going to secrete proteins, metabolites, and secretory vesicles, metabokines, that are going to travel through the blood and arrive to different tissues. During this trip, they are going to target liver, kidney, and even brain, and they will affect the functionality of this organ, indicating that there is a problem in the adipose tissue. Among all of them, in the heart, these metabolites are going to signal and disturb the heart function. This scenario leads to ventricular dilatation, increased ventricular wall stress, compensatory hypertrophy, and diastolic dysfunction, resulting in congestive heart failure. Adipose Heart will study how adipose tissue can contribute to this effect through the secretion of metabokines.
And I want to finish thanking all the people of my lab, the people at the CNIC that have helped us during this year, and the people outside, including Alejo and other people surrounding us that will help us all the days in our research. Thank you very much.
Okay, thank you, Juana, for a fantastic presentation. I'll remind the people that the Q&A tab that you can see at the bottom right is the where you can type your question, and I will read them out loud so that what I can answer. But before that happens, let me start myself by asking you a few questions. So there's a lot of functions that different isoforms under different circumstances this stress kinases have, and how do you see both in terms of selectivity and in terms of indication, together with side effects, that the pharmacological targeting of the stress kinases might operate in the clinic?
I think until now, most of the inhibitors have been that the ones that arrive to the clinics have been the more spectrum ones, like the inhibitor of ASK1, that is the upstream of the P38 and JAKs. At the end, this type of inhibitor have secondary effect because you are going to inhibit all the pathway. more potent, but also the effect are worse. That means I think we need to use more specific inhibitor, perhaps targeting them to one tissue, for example, with peptides. There are now pharmacological ways to make that one inhibitor will arrive mainly to one tissue and not to other. And I think that is going to be one thing. And the other thing is to try to do inhibitors to a single kinase and not to the amplius spectrum kinases.
Mm-hmm. And how far are those selective inhibitors from the clinic?
I think there are some that are in preclinical steps. However, I think we are still far away to have a good inhibitor of these kinases, I think.
Mm-hmm. Mm-hmm.
I think we need to develop or more better ways to do an inhibitor. The problem with inhibiting a kinase is that the active center is very similar in all of them. That means make very difficult to have a inhibitor specific for one or another. I think we need that to get into the clinic.
And regarding that one, another important aspect is that you know, by playing with these in mice, so you know, they have different functions. You actually showed the effect of JAK1 and 3 opposite effects in controlling food intake in the central nervous system. How is that dictated from this molecular point of view? Is it because of the upstream modifications, or is it because of the selectivity towards targets? Because of course there has to be, you know, when you have so many upstream signals and so many downstream effects, that there has to be an input dependent activity, right? You can't have just it bluntly activated and you know, phosphorylating whatever targets regardless of the input, right? So how is that at the molecular level controlled? It's more regarding the upstream activation, regarding the selectivity of these different isoforms?
I think it's both. What was described from the beginning was that all of them are activated for the same upstream pathway, but what we found now is that is not true. Depending of the tissue, there are upstream activators that is the main upstream activator, and the other ones only work when the other one is not there, or as a helper, but not the main upstream activator. And for example, in heart, we know that P38 are controlled differently, P38 alpha and beta, than P38 gamma and delta. In this scenario, I think it's also very important scaffold protein that are going to help them to signal, and perhaps targeting this scaffold protein that perhaps are more specific will help the clinic to know which scaffold protein binds to each of the isoform and which one are important in what one signal versus other.
No, I see. And so going back to this, you know, these kinase effects, and so you've learned a lot using knockout mice, right? But that's not completely different, but it's different than inhibiting a kinase. So yeah. Is there any evidence that there's no kinase effects of the stress kinases?
I don't think there are evidence of that the kinase doesn't work without all their activity is through the kinase activity, and they don't have any other function like a scaffold or helping each other. In fact, it's clear that inhibition of the kinase is not exactly the same that knocking the kinase, because these kinases, I think, have also some scaffold function that when we have a knockout, we avoid this function. We deplete all the function. For example, for trying to understand that, we are starting to block the upstream kinases to have the like, for example, the P38, but not the P38 active, and to see if that is the same that knocking out the kinase, because that will help you to understand what is the real function as a kinase and the function as a scaffold protein or a pseudokinase.
Well, correct me if I'm wrong, but do you know that the levels of the different isoforms are affected by the deletion of some of them, right? So,
yeah.
So that's likely to show some differences.
Yeah, when you, for example, P38 alpha is going to control not only the transcription, but also the RNA degradation of the other P38s. Also P38 alpha is going to control the JAN activation. That means when you deplete P38 alpha, you are going to affect all the other kinases. And for that reason, some of the deleterious effects of P38 alpha inhibitor are due to this hyperactivation of the other stress kinases.
Mm-hmm. Mm-hmm. Because there's a lot of points knocking mice using CRISPR that are worth making. Then,
yeah, I
think
so.
enzymatically dead CRISPR mouse if they live, right? But,
yeah,
but I guess there's a lot of work to be done there. Then.
I think knocking the active side will help us to understand a lot of the kinase function versus the no kinase function.
Mm-hmm. I see. Good. So I'm checking here. I don't know if I'm looking at this properly. There's a question here from Maria Caparel. Hi, what a great talk. I love the video as well. Yeah, me too. Are the stress kinases always activating the same cytokines, e.g. IL-6, or does it depend on the organ context? I'm asking because it looks like one main target of stress kinases is always IL-6. Thanks, Maria Caparel.
Well, P38s are very important regulator of interleukin-6 and TNF-alpha, mainly in macrophages. Also have been described that JAN control interleukin-6 in adipocyte. I don't think in all the organs are going to control interleukin-6 production and TNF-alpha production, but also these kinases are activated by this cytokine, like it's an internal cycle that macro activation of them in the inflammatory cells are going to induce interleukin-6 and TNF, and then this interleukin-6 and TNF is going to activate stress kinases in the other tissues, and that make the signal to maintain during the disease. That I think that is one of the important problem of a chronic inflammation, that you should have the stress kinases most of the time sleeping, but with this chronic inflammation, you have these stress kinases in a constant activity.
In 5 minutes, the joint area seminar.
Okay, Joanna. So I don't see any other question. I guess that some of the people might be willing to stay on the second half of the session, so that they can directly interact with you and asking questions on your presentation or anything else related with science that they might be willing to know from you. So Rita, I'm not sure how to do this, but I'll give a closure to this first part of the session, thanking all the attendees and thanking EMBO for organizing this, and of course Guadalupe, Sabio for a fantastic talk. And those who are willing to stay and talk to Wada directly, I think should stay, and we'll be staying here, I guess. So thank you very much.